Everyone's body has an immune army patrolling 24 hours a day. T-cells are like special forces, circulating to identify and eliminate invading pathogens and abnormal cells. However, cancer cells are very cunning. Through mechanisms such as genetic mutations and antigen modulation, they cleverly disguise themselves, evading the immune system's routine monitoring to proliferate freely in the body. Traditional treatments, such as chemotherapy and radiotherapy, are like indiscriminate bombing, damaging healthy tissues while targeting cancer cells. Facing these limitations, the medical community has turned its attention on the body's own arsenal, the immune system. By genetic engineering, we equip the body's immune cells, T-cells, with a GPS navigation system to target cancer cells. The modified T-cells are upgraded to CAR T-cells, capable of precisely locating and killing cancer cells. This therapy not only breaks through traditional treatment models, but also brings hope for long-term survival in advanced cancer patients, opens a new chapter in precision cancer therapy. T-cells play a critical role in the human immune system, which can directly kill abnormal cells. T-cells rely on MHC molecules to recognize tumor antigens. However, the lack of MHC molecules allows tumor cells to evade immune recognition and attack through various mechanisms, leading to immune escape and suppression of T-cell function. CAR T-cells can specifically recognize antigens on tumor cells. This recognition is independent of MHC, overcoming immune escape caused by the lack or reduction of MHC, enabling precise targeting of tumor cells. When CAR binds to tumor antigens, CAR T-cells are activated, initiating intracellular signal transduction pathways that promote cytokine secretion, further enhancing T-cell expansion and activation. Activated CAR T-cells multiply in large numbers, forming a powerful cell population, which directly kill tumor cells. The core of CAR T is genetic modification. The treatment only needs 5 steps. First of all, draw on the patient's blood. The blood contains the key members of the immune system, T-cells. Next, T-cells are extracted from the blood for genetic modification. Gene modification is the core step of CAR T-cell therapy, using viral vectors. The artificially designed Chimeric Antigen Receptor CAR is implanted into the T-cells. The modified T-cells are cultured and expaneated to reach the quantity required for treatment. Once the CAR T-cell amount meets the standard, they are reinfused into the patient. They can quickly target tumor surface antigens, launching a fierce attack on tumor cells, and continuously engulf and destroy tumor cells, thus achieving the goal of treating cancer. Throughout the process, the medical team closely monitors the patient, managing any adverse reactions to ensure the safety and effectiveness of the entire treatment. CAR T is a revolutionary force in cancer treatment. Currently, 12 CAR T therapies have been approved globally, bringing hope for patients with blood cancers such as leukemia and lymphoma. However, solid tumors, which account for 90% of cancers, are facing challenges. NG Biomed has always been committed to technological breakthroughs, with its dual innovative strategy based on the CRISPR-Cas9 gene editing technology, opened up a new path for the treatment of solid tumors. In the tumor microenvironment, cancer cells inhibit T-cell activity by binding PD-L1 molecules to PD-1 on CAR T-cells. NG Biotech uses gene editing to knock out PD-1 in CAR T-cells, significantly enhancing their sustained tumor-killing ability. MUC1 is a tumor antigen widely expressed in solid tumors, such as lung and breast cancer. In normal cells, MUC1 is shielded by intact glycans, but in cancer cells, glycan defects expose MUC1 abnormally. CAR T-cells designed to recognize this tumor fingerprint to avoid harming healthy tissues. NG Bio's dual innovation CAR T-cell therapy has 4 advantages. CAR T-cells target tumor antigens by genetically engineering the patient's own T-cells, overcome immune escape of tumor cells to accurately identify and eliminate cancer cells. The new generation of CAR design gives CAR T-cells immune memory, allowing them to survive in the body for a long time and sustain suppression of tumor recurrence. Aiming at abnormally exposed MUC1-TN glycoepitopes on cancer cells, accurately distinguish cancer cells from normal cells, significantly improves treatment, accuracy, and safety. CAR T therapy designed for antigens with high expression across different cancer types can treat lung cancer, breast cancer, esophageal cancer, and other solid tumors, significantly expands the indication range. A 47-year-old male diagnosed with advanced lung cancer with widespread bone metastases and inoperable conditions was given an expected survival time of only 3 months. After multiple CAR T treatments at NG Biome, his survival extended to over 32 months. The lung lesions shrank significantly and did not recur. His quality of life improved dramatically, from limited mobility to live and work independently. CAR T therapy is highly personalized. NG Biomed tailors treatments based on the patient's immune system and tumor characteristics for precise cancer cell targeting. The future of CAR T-cell therapy is full of possibilities. As a global leading biotechnology company, NG relies on 4 core technology platforms, including high-yield virus, recombinant antigens, gene editing, and cell preparation. At NG, technological innovation will continue to drive breakthroughs, advancing toward improved efficacy, reduced costs, and minimized side effects. Looking ahead, clinical application has a bright future, while consolidating successes in blood cancers and autoimmune diseases. Solid tumor treatment may soon see breakthroughs. With the maturity of technology and cost reduction, CAR T-cell therapy will continue to expand its market and gradually becomes the standardized treatment for more diseases, bringing revolutionary changes to human health.